Good afternoon, everyone. Today we'll be learning about biomolecules. Contents in this lesson are types of biomolecules, amino acid classification, nucleic acid classification, structure of DNA and RNA, biological functions of nucleic acids. A definition for a biomolecule is biomolecules are complex organic molecules that play an important role in the structure, function, and regulation of living organisms. The four major types of biomolecules are carbohydrates, lipids, nucleic acids, and proteins. They are important for the survival of all living cells. They are mainly composed of the elements like carbon, nitrogen, hydrogen, sulfur, oxygen, and phosphorus. They are vital for life because they help the organisms to grow, sustain, and reproduce. Examples for carbohydrates are starch and cellulose. For lipids, fats, oils, and waxes. For nucleic acids, DNA and RNA. And for proteins, insulin and collagen. Carbohydrates are useful for energy source and they store reserve food. Lipids are also for energy source and they are membrane component. Nucleic acids will store genetic information, and proteins, they form the enzyme structure, helpful in movement, and the oxygen carriers. Amino acids. Amino acids are small molecules that are the building blocks of proteins. They serve as structural support inside the cell and they perform many vital chemical reactions. Each protein is a molecule made up of different combinations of amino acids. Amino acids help in the breakdown of food, growth, and repair of body tissue. They make the hormones and the brain chemicals. They are a rich energy source. They maintain healthy skin, hair, and nails. Coming to the classification of amino acids. Amino acids are organic molecules that are composed of an amino group and a carboxylic group with a specific site, that is R group. Classification of amino acids. Amino acids can be classified into two types based on R groups, based on the ability of synthesis of organism. Based on the ability of synthesis by organism, again there are three types: essential amino acids, non-essential amino acids, and conditionally essential amino acids. First one, based on R groups. These amino acids can be synthesized based on the R groups attached to the side chain. These R groups can be polar or non-polar. They can be called aliphatic amino acids or aromatic amino acids if the R group is aliphatic or aromatic. They can be classified on the solubility of water also. If the R group is hydrophilic, then it is called as water soluble, and if it is hydrophobic, it is water insoluble. Every amino acid is represented as a three-letter abbreviation and with a single letter symbol. The next one, based on the ability of synthesis by organism. First one is essential amino acids. These amino acids cannot be synthesized in the body. There are no special metabolic processes inside the body to synthesize these essential amino acids. They do need to be supplemented via diet externally only. There are 9 such amino acids. Examples for essential amino acids are methionine, threonine, histidine, phenylalanine, valine, isoleucine, tryptophan, lysine, and leucine. Non-essential amino acids are the amino acids that can be synthesized by the body. There are metabolic processes inside the body to synthesize these essential amino acids. So we need not supplement them diet. Examples for non-essential amino acids are alanine, asparagine, glutamic acid, and serine. Conditionally essential amino acids are synthesized by the body but can become essential amino acids due to some pathophysiological conditions. So they become conditionally essential and need to be supplied through our diet. There are 6 such examples for these amino acids. Examples are arginine, cysteine, glycine, glutamine, proline, and taurine. Nucleic acids. These nucleic acids are large biomolecules that are crucial in all the cells. They are biological macromolecules essential for the storage and transmission of genetic information. There are two main types of nucleic acids: DNA, that is deoxyribonucleic acid, RNA ribonucleic acid. Nucleic acids are composed of smaller units called as nucleotides. Each nucleotide again consists of three components. First one is phosphate group, second one is a sugar molecule, deoxyribose in a DNA and ribose sugar in RNA, and a nitrogenous base. The nitrogenous bases again are classified into two types: purines, which consists of adenine and guanine; pyrimidines, which consists of thymine and cytosine in DNA, and uracil and cytosine in RNA. The sequence of these nitrogenous bases will encode the genetic information in the form of genes, guiding the synthesis of proteins and other cellular activities. These are the structures for nitrogenous bases, purines and pyrimidines. Deoxyribonucleic acid. DNA is composed of a pentose sugar, a phosphoric acid, and some cyclic bases containing nitrogen. The sugar moiety present in DNA molecule is beta D 2-deoxyribose sugar. The cyclic bases that have nitrogen in them are adenine, which is abbreviated as A, guanine G, cytosine C, and thymine T. These bases and their arrangement in the molecules of DNA will play an important role in the storage of information which is passed from generation to the next generation. DNA has a double helical structure in which the strands are complementary to each other. Coming to RNA, ribonucleic acid. The RNA molecule is also composed of phosphoric acid, a pentose sugar, and some cyclic bases containing nitrogen. RNA has beta D ribose sugar in it. The heterocyclic bases present in RNA are adenine, abbreviated as A, guanine G, cytosine C, and uracil U. In RNA, the fourth base is different from that of DNA. The RNA generally consists of a single strand which sometimes folds back, that results in a double helical structure. There are three types of RNA, each with a specific function: messenger RNA called as mRNA, ribosomal RNA called as rRNA, and transfer RNA called as tRNA. Coming to the structure of DNA, DNA structure is thought to be as a twisted ladder, but it is described as a double helical structure, which is shown in the next slide. It is a nucleic acid, and all nucleic acids are made up of nucleotides. The DNA molecule is composed of units called nucleotides, and each nucleotide is composed of three different components such as sugar, a phosphate group, and nitrogen bases. The basic building blocks of DNA are nucleotides, which are composed of sugar group, a phosphate, and a nitrogen base. The sugar and phosphate groups link the nucleotides together to form each strand of DNA. Adenine, thymine, guanine, and cytosine are four types of nitrogen bases. These four bases together pair in the following way: adenine with thymine and cytosine with guanine, A with T and C with G. These base pairs are essential for the DNA's double helical structure, which will resemble a twisted ladder, also called as double-stranded DNA. The order of the nitrogenous bases will determine the genetic code of the DNA's instructions. This is the component of DNA structure with the phosphate group, nitrogenous base, and deoxyribose sugar. Among the three components of DNA structure, sugar is the major one. It forms the backbone of DNA molecule. It is also called as deoxyribose sugar. The nitrogenous bases of the opposite strands form hydrogen bonds, forming a ladder-like structure. This is the structure of DNA, which is a double helical structure. Structure of RNA. Ribonucleic acid also has the same components of DNA with only two main differences. It has the same nitrogenous base called the adenine guanine cytosine, and thymine is replaced with uracil. Adenine and uracil are considered as the major building blocks of RNA, and both of them form the base pair with the help of two hydrogen bonds. RNA resembles a hairpin-like structure, and like the nucleotides in DNA, nucleotides are also formed in this RNA material also. Nucleosides are nothing but the phosphate groups which sometimes also help in the production of nucleotides in the DNA. This is the structure of RNA molecule. Coming to functions of RNA. RNA, which are mainly composed of nucleic acids, are involved in a variety of functions within the cell and are found in all living organisms, including bacteria, plants, viruses, and animals. These nucleic acids functions as a structural molecule in all cell organelles and are also involved in the catalysis of biochemical reactions. The different types of RNA are also involved in various cellular processes. There are various types of RNA, but well known are tRNA, which is called as transfer RNA, rRNA, ribosomal RNA, and mRNA, messenger RNA. Transfer RNA. This is responsible for choosing the correct protein or the amino acids required by the body, in turn which helps the ribosomes. It is located at the end points of all the amino acids. This is also called as double RNA, soluble RNA, and it forms a link between the messenger RNA and the amino acid. Coming to ribosomal RNA, it is the component of the ribosomes and are located within the cytoplasm of a cell where ribosomes are found. In all living cells, the ribosomal RNA will play a fundamental role in the synthesis and translation of mRNA into proteins. The rRNA is mainly composed of cellular RNA, and they are the most predominant RNA within the cells of all living beings. Coming to messenger RNA, this type of RNA functions by transferring the genetic material into the ribosomes and pass the instructions about the type of proteins required by the body cells. Based on the functions, these type of RNA are called as messenger RNA. This mRNA play an important role in the process of transcription or during the protein synthesis. Biological functions of DNA and RNA. DNA is responsible for maintaining the identity of different species of organisms over the past million years. It is a chemical basis of heredity and is also considered as a reserve bank of genetic information. It directs the synthesis of RNA, which in turn directs protein synthesis. DNA is required for replication and heredity. Various cellular functions are under the control of DNA. A DNA molecule is also capable of self-duplication during cell division, and identical DNA strands are transferred to the daughter cells. DNA controls the synthesis of RNA. The sequence of these nitrogen bases determine the protein development in the new cells. DNA holds genetic information, whereas RNA transfers genetic information from DNA to the ribosomes. RNA facilitates the translation of DNA into proteins. RNA also functions as an adapter molecule in protein synthesis. RNA serves as a messenger between the DNA and the ribosomes. RNA are the carrier of genetic information in all the living cells. RNA promotes the ribosomes to choose the right amino acid which is required in building up of new proteins in the body. Thank you.